Good morning. It is good morning. I like that. It is 9:30 on the dot, which means it's Saturday 9:30. It is time for another Science on Saturday. I'm so glad to see everybody here. We held the snow off till tonight on purpose just to make sure that there was no issue with the lecture. Anybody here for the first time? You've got a few, but you've got a lot of veterans here today. Well, you already know, welcome back. For the first timers, we know that this is part of the reason why, of course, you are here, and we will continue to make sure you are well fed and there's coffee for everybody. Of course, those who are here and those who are on Zoom, we'll hold our questions until the end, and then we'll pass microphones around to make sure that not only you can hear the question in the audience, but our folks online can hear it. And before I introduce our speaker, I mentioned quickly that the March lectures are all going to happen on campus. We've confirmed that. We're working out the details, but like I said, it will be on campus with a parking garage right more or less next to it. It is part of the Anlinger Center, for those of you who know campus, right off of Prospect Avenue, right downtown. So just a few miles from here. Parking garage, we'll make sure all the signage and all the directions are both online, and once we have it all finalized, the logistical part here, we'll make sure it's here as well. But that'll be the 3 lectures in March. So, say again? Oh, the question was, will there be donuts? I have good news, which is, if you think that we were going to move to campus and not bring the donuts, then we are well aware. I will say one little bit, just to give you a little foreshadowing. The donuts can't come into the auditorium, and so we're going to have to do all of our eating and drinking in the lobby outside of that auditorium before we come in. But if you can handle that, then we're good. Well, I am delighted to introduce Professor Corey Toler-Franklin from Columbia University to Science on Saturday. You can see her incredibly wonderful and impressive resume here. She is, of course, we are welcoming her back as well as a Princeton graduate. Columbia, Princeton, she's been at the University of Florida, she's won a bunch of different awards, she's worked in this wonderful intersection between graphic design and computer science, and her lecture today is Quantum Level Spectral Analysis and Deep Learning for Life Science and Biomedical Research. Would you please welcome Professor Toler-Franklin?
Thank you for being here. Hi.
You are in the middle. So before we get started, of course, I have to ask you 3 questions. The first question is, tell us, when did you first get interested in science?
So this all started when I was an undergraduate researcher at a lab at Cornell University. And this lab was at the forefront of computer graphics at the time. And it was just a wonderful experience to be a young person interacting with the biggest names in computer graphics at the time. And it was also, I was an architecture student, and it was at the time that technology was being introduced to architectural design. So I got to see these realistic image synthesis algorithms using Monte Carlo sampling and all these complex things being used in my field. And so I thought, could I, use that? And I was also frustrated with the way technology was used in architecture. So I thought, could I do research to change that? And that's how I kind of got on this path. I graduated early, did a master's degree focusing on computer science and graphics, and that's how it all started.
So given all of that, tell us about a teacher who had an influence on you, whether that is PhD, master's, undergrad, or kindergarten, or anywhere in between.
So that'll be Donald Greenberg, the person at my crossroads. So Donald Greenberg was the person who recruited me for that lab, and he has this engineering architectural background, and he had made great headways in computer science. And the 3 reasons why he's important. The first is that he saw in me my potential before I saw it myself, and he knew that I could do this math and science. The second thing is that he gave me an environment. I was born in Florida, but I grew up in the Bahamas. So here I was, I met him at 15 at a summer explorations and architecture program, and I took his structural engineering class, and he provided an environment where we had the resources, the funding, everything we needed to be successful. And the third reason is he's extremely brutally honest. If my stuff didn't work, it didn't look right, he had no problem telling me. And I think this is important as professors because my growth came from his honesty. And yeah, so Don, for sure.
That's a wonderful story. So last one is when you're not doing your research or you're not teaching, something that you do for fun.
I play the piano. I thought that I was going to be a pianist. That's my first my first thing. That was my dream, and I took this side road into architecture. But playing for me, I believe that it's part of my creativity. It helps me be creative, and also it makes me very happy. So when I'm stressed out about an algorithm or something, I sit and I play, and I have a spinet that's from 1942, antique, and I have an 1880 Shallon, and so I love pianos.
And with that, quantum level spectral analysis, deep learning for the life sciences and biomedical research by Professor Toler-Franklin. Thank you so much.
Thank you. Okay, so today what I want to do is I'm going to tell you a little bit about my lab. This is gonna be a broad talk about the type of research that we do, and then I will get into using deep learning, and we'll talk about what I mean by deep learning. So deep learning for biomedical research, and I will focus mostly on the cancer research. And then I'll show you some motivating problems in life science where we looked at some spectral analysis and give you a sneak peek into how we're expanding that for forensics with some field work at the site of the Tulsa Race Massacre of 1921. So my research is at the intersection of computer vision, graphics, and optics. So from computer vision, we're looking at how we can build sensory devices to capture the world around us as we see it. And then from a computer graphics standpoint, developing rendering algorithms or using AI for analyzing that data. And here I'm just showing you some images from my lab. So this is my first lab at the University of Florida, and I've kind of lifted all that up, and it's now at Columbia. And so what we're doing is looking at algorithms that build upon principles of optics and physics. So we're looking at spectral imaging, 2D, 3D scanning, reflectance spectroscopy, microscopy. So we actually customize these devices and develop different algorithms. And I cannot go any further without telling you where it all started. So my thesis at Princeton actually was going on site at places like this archaeological dig, and figuring out how we can capture these data, these large volumes of data, like these fragments of frescoes that you see here, and then looking at practical ways and practical algorithms to get some use out of it. So here we're looking at computer-assisted matching. And I was really enamored by the idea of having images that had more than just color. So here you're seeing images that have 3D orientation. So everything that's pointing towards you is blue in that second half. To the right is red, up is green, and to that kind of indigo colors to the left. And what we can do is we can develop algorithms like this where you don't see details in that color image, but as soon as you start doing these enhancement algorithms, you can see like the etching on the surface of that tool, or you can see the manufacturer's tag actually on the barrel there that you just don't see in that image. And all of the work that you see here is born out of my experience kind of saddling between industry and academia. So, so you've seen my profile. I've been at all these places, but then I also worked industry, and I looked at challenges of looking at real world problems that real professionals face, but then using it to fuel core problems in computer science. And so this means that I have to have a lot of collaborations. I'm appointed at the American Museum of Natural History. I have collaborators at the Paleontological Research Institute in New York, and I'm also have an affiliate appointment at the University of Florida in the museum there. And I was a part of this iDig Bio fellowship where we're looking at digitizing all of the nation's biodiversity. And that's how I got into the types of projects I'm working on. And so when you come to my lab, you'll see that there are four main projects. AI for cancer detection, looking at biomarkers and detecting them. AI for neuroscience, how can we look at phenotyping animal behavior for diagnosing neurological disorders? Quantum physics for appearance modeling and being able to model materials, and AI for forensic science, and that's this cultural awareness and cultural understanding project. But the unique thing about my lab is when you're sitting there, you'll see the graduate students working on their computer science projects, but you will see researchers from other collaborators or from other fields working together. So for every one of these projects, there's a core computer science problem that you see on the right in green, whether it's vision transformers or looking at multispectral rendering or looking at social network reconstruction. So it's a very dynamic environment. And this spews into the courses that I teach. So I introduced a new course at Columbia this semester that looks at neural rendering for computer graphics, taking the latest in neural networks and this field that's really bustling at SIGGRAPH, which is our major conference, and looking at methods that use neural rendering to solve some complex problems in graphics that were computationally intensive before. And so there are a number of my courses that combine deep learning with computer graphics. And I also taught a course at the medical school for medical honors students, looking at how they can take these techniques and incorporate it in their fields. And a lot of the projects have arms into education. We're looking at STEM education and putting 3D printing and capture in the classroom. And I don't do this alone. This is my team. And so these are the four teams in my lab, and the ones with the red are the lucky ones who are now at NVIDIA or AMD and doing really great things. Tan just graduated with his master's from Columbia, and he's at NVIDIA now. So let's start looking at this deep learning. So we know that artificial intelligence, a neural network is a network that's inspired by some biological network, and that is very loosely said, which are used to estimate or approximate functions. And so we want to depend on a large number of inputs, and we generally don't know what we have, and we want to be able to somehow classify or work with that data. And the idea of machine learning was really first coined by Arthur Samuel, who's looking at a chess game. He has this chess game where for the first, not chess, checkers, where for the first time he's actually having an AI play this game. So this is a field of study that gives computers the ability to learn without being explicitly programmed. And there are three categories within this. Supervised learning, that's when we actually label our data set and we try to learn so that given a new data set that we don't know, we can figure out a label. Or we can do something completely unsupervised where you give the network an image, and because of the actual structure of that network, it can figure out the structure of the data. And then there's reinforcement learning, where we have some agent working and changing in an environment, and you're trying to maximize some reward for that agent. So when I say deep learning, I'm talking about algorithms that span these three areas, but the unifying thing is they all use neural networks. So let's look at that idea of supervision in terms of this project with AI for neuroscience. So in neuroscience, animal behavior phenotyping and actually analyzing behaviors in videos is at the cornerstone of a lot of the research that we do in human clinical studies. But one of the problems is that a lot of that information that's learned isn't transferable or doesn't transfer a lot of times into these clinical studies. And there are a number of reasons for that. And one of the reasons we focus on is the challenges with this manual scoring. So a neuroscience would manually label videos with behaviors, and then we have to somehow use a network to classify. But the challenges are that you can imagine me, I'm learning how to score. There are variabilities between when I learn to score and my maturity over time with that task. There's variability across research labs, so how do you reproduce this? And there's human bias, and these methods are time intensive. So here you're seeing an example from deep ethogram where you're seeing these different behaviors being labeled, and you have the ethogram on the bottom that's basically telling you for each frame in that video, what is the behavior that's happening. And so we put out this survey paper where we looked at how people were implementing these animal behavior analysis. And what we found was, if you look here, this is just showing all the different networks that are used for human action recognition and human pose estimation. And then we overlap that with methods that are used for animal behavior classification. And we found that the human pose estimation and action recognition methods were much more mature than those used in animal behavior analysis. And so we found another interesting thing. If you look at the ones that are blue versus the ones that are that pinkish color, the blue ones claim to be unsupervised. If you want to do this, you want to be unsupervised. You want to have a video, and you want to automatically tell you what's going on. It also means that it can tell you micro behaviors that a human may not have thought of or may not have put in a library of behaviors. So that's what we want. But what we found was that even methods that claim to be unsupervised still relied on some manual labeling of data. And so here, this is a supervised method, and there you're seeing there's your manual labeling. A network, it does its stuff, and then at the output here, you put some new video in, and it's going to be able to tell you what the labels are based on what it learned from a data set of videos on the left. But here's an unsupervised method that came out, I believe, might have been 2019 or so, but this method says it's completely unsupervised. However, it is unsupervised at that end, but it relies on hand-labeled data from pose estimation. So that is an issue. Another issue is some of these methods that are unsupervised have a lot of vision, hand-tuned processes, whether it's pre-processing of that data, some computer vision technique. So in our lab, what we're doing is first we presented a taxonomy that classified these methods based on the level of supervision, and we're now going to be publishing a data set and a method that uses adversarial learning as a step towards a more unsupervised approach. So now I'll go into a little bit of more detail with our cancer detection project. So here is something that we can look at. So these are the types of data that we analyze. And our goal is to find the tumor in those slide scans of tissue. And you'll notice that there's several problems. The biggest one is the variation in that data. So here on the left, you're seeing data that's like really, you have this really big tumor, but then you can have some tumors that are like microscopic in size, a few nanometers. So how do we deal with that? Another big problem is the fact that they're non-discriminative features. They're features that tend to blend into the background. And so in the bottom there, it's hard to decipher. And the actual process of labeling this in a lab and then having an oncologist go and look at it and evaluate it is time-consuming. And so to understand why this is a problem for a network, let's think about what we mean when we learn from data. When we learn from data, you take a task, you want to repeat it a lot on a lot of data, that's your experience, and then you want to somehow get a performance metric or some performance measure of that. So Goodfellow says a computer program is said to learn from experience E with respect to some class of task T and performance measure P if its performance at task in T as measured by P improves with experience. And the problem or the principles that we think about when we think about AI have to do with how is this learning happening? Am I going to sit down at a computer and program a robot to say, here's how you move, here's your first step, your second step? Or am I going to give it a bunch of data, show it how humans move, or maybe how I want it to move, and let it learn how to do this? This is the difference when we talk about artificial intelligence. And every one of these algorithms have a data set, and so there's a wide range of types of data that you deal with, and there are issues when you have sparse data. They all have a model. So in this case, I'm showing you an example of an image classification task. So in image classification, we're going to use our neural network to learn some function or some set of parameters. You're going to learn those parameters that are going to help you to map from some initial space X to some other space Y. So my image is in one domain. My classifier, that's going to tell me what class it is, 0 through k minus 1, is in another. And so we're mapping from k to some target domain that we're looking at. And so at the end of all that, then we have to do some optimization. The name of the game with neural networks, you're going to minimize some function to figure out if you're performing well. And so here you're seeing in this graph, 1, 2, 3, 4, these are the different inputs X, and they all represent different classes. So if we're looking for the probability that you have class Y given X2 at that first line, and you're seeing the values intersecting the graph. And here we're looking at the discrete probability Y given X is 7, looking at that graph. So now that I gave you some background just on how neural networks work, why is this challenging? Why is my data set challenging? Well, one problem is if you look at how a convolutional neural network works, it takes in your image, it does a process of averaging called convolution, and it does this on a very small resolution image, and then you get a higher detail at the end. So just the process of operating on such a small resolution image versus all the data and information you have in your high resolution, what happens? How do we capture that? Another challenge is, can anyone see what's in there? Right? So I've zoomed it in for you. I've zoomed it in so you can see this. So imagine you have the whole big picture. It's hard to see, but one of the challenges with our medical data set is, well, the concealed spider looks the same as the background when you look at these patches. So how is our network going to distinguish that, particularly when we look at our cancer? A cancerous patch can look the same as another cancerous patch. And so the networks that we use to do this are transformer networks. So transformer networks are the crave. They're really cool because you can think of them as being able to find long-range relationships between time series data. So what you're seeing here is in A, you can think of X1, X2, and X3 as words in a sentence. So your words are going in. And then for every output that you see, there's going to be some weighted relationship with all the other words in the sentence. And the dot self-attention module is going to compute something that's going to tell you help you to actually classify what that word is. And so as we move from left to right, every time we switch to our next word, it's going to be a different set of weights that are going to help us to figure out what that word is in the sentence. So I put this visualization here because I want you to use it to understand what problem we're trying to look at. Here, the network is firing some information that tells us it's picking up some differences in our words. So there's a self-attention block to the left, and we compute 3 values: a query, a key, and a value. You can think of queries as adjectives in the sentence. So Corey's wearing a blue suit. That blue is your query. The key is actually going to give you the answer, whether when you're in this big space, if you found the word that you're looking for, and the values are the weights that we saw before. So what the main issue is that if you have words like xi and xj that look similar, their queries are going to look similar, their values are going to look similar, you're going to get over smoothing and you can't figure out what's going on. And so let's look at how we actually worked on a project to solve this problem, or some of these problems. So this is a collaboration with an oncologist at the University of Florida, Goy Helderman, and we want to be able to find the tumors in this data set. So the first thing we do is we have a strategy. We're saying that the challenge is that there are a broad range of sizes, they're non-discriminative, it's time-consuming. So how are we going to address that? Well, the first thing we're going to do is we're going to make sure that there are multiple detection layers. So we're going to make sure that in each layer of our model, we have different scales that we can detect at each of those layers. We're going to make sure that we can get background information as well as local context. And the term we use, we have a cascade residual inception module and deconvolution. That's what we actually do. But what we're doing is we're finding broader information beyond just color or image space information. And we're going to do it in a single shot. We're going to just do one quick pass to do this. So how do we actually measure the performance of how our network works? This is a measure that we use. It's called intersection over union. So that green box you see there, that's the box that the model predicted. The red is actually the ground truth. So the ground truth is obviously tighter around the beta fish, where it's correct. But on the left, you see that this measure, this is the area of the overlap over the area of the union, is very poor. The value is closer to 0. And that's because it has too much of the background information there. All the way to the right, it's closer to 1, which is where we want to be because the overlap with the ground truth is correct. And then you have variations in the middle. This slide is important because I'm going to talk about how we predict priors over our data. Priors are those ground truth boxes. We want to be able to predict a set of boxes over our data that's going to be as close to those ground truths as possible. And so here are our prior anchor boxes. So these are little boxes that we compute. So we want to pre-compute these. We want to be able to match the distribution of where tumors are in our data set. And then we want to be able to see if we could detect. So what are we going to do? The first thing we do is we're going to make sure we have a better chance of finding the tumor. If you look at that pyramid, that's what the images look like at different layers in that network. So further away, you see that you have a larger resolution and the actual tumor covers a larger part of that image. As you move up in that pyramid, that portion becomes much smaller. So in our network, we actually use this single-shot multi-box detector, which is a method that has these prior anchor boxes that are fixed at each layer. But what we do is we add more higher resolution images to it, so we have a better chance to detect our tumors. Another thing we do is we change the ratio and the sizes of those anchor boxes dynamically so that we can detect a larger range of data. And we have double sets of these to have a better chance of doing it. So SSD, which is this first method of doing this, doesn't work that well on microscopic data because it doesn't have these dynamic features. And so this is just showing that we're going to make sure that when we compute these anchors, that they should be a small amount, but cover as much of the ground truth as possible, and that we have a proof in the paper that determines how we actually compute the aspect ratio. And so in the end, we have, these are the different layers of our network. So 1, 2, 3, 4, 5, we have different layers there. Stride is when we do convolution, how we move along in our network. And the anchor size, we're looking at the size of those boxes and the aspect ratios that we compute and the effective region that we can actually see. So here's a good visual of how it works. On the left, you see the model that we build upon, and on the right, you see our approach. The y-axis shows the height of the actual detection region for our anchor box, and the width is shown on the x-axis. Each of those lines that are just shooting out, the aspect ratio is the same. Along those lines, they can be different sizes, but the aspect ratio is the same. And the red are our first computed boxes, and the green are our second, and the big cloud, black cloud you see, is the distribution of tumors in our data. And so you can see on the right, we actually are able to compute more of those prior anchor boxes and more likely find where the tumors are because we are closer to the denser region there. And so I won't go into details of the network, but some of the key things is that our first network just does regression and classification, and we do something more complex. We add more detection anchor boxes with this inception layer, and we can get non-discriminative features. And there's lots of details of things that we do to speed up the learning. And then we add this yellow deconvolution layer to get context from around. So here's an experiment that we did. We have 30,000 patches extracted from microscopic scans. Each patch is 300 by 300 pixels. We have these tumors that could be 8 pixels all the way up to 300 pixels in size. We separate our training and test sets. This was done on a bit earlier. We can do this much faster with the latest GPUs that we have now. And here we're doing 180 iterations of this training and 100 iterations of fine-tuning because we're fine-tuning a network, our VGG network, to do this. And here's what we found. I'm comparing it against methods that do 2 passes for classification. So they first do a search, figure out what they're detecting, and then classify. I'm comparing with SSD, which is our method, where we actually do it in a single pass, where we do classification and search at the same time. Then 3 different additions that we have added. This adaptive prior anchor boxes, this foveal residual inception that gives us the discriminative features, and then this deconvolution, which gives us more background context. And we're looking at precision and recall. So precision, once we have our tumors detected, this is the fraction of tumors that we've detected that are truly cancerous tumors. When we do recall, it's how much that we've detected so far compared to how much we should have detected so far. And then we have an average of the precision on the right. And so what we found is the 2-pass methods, precision's pretty low, but we get about the same precision as SSD. But then when we look at recall, the 2-pass methods recall a lot of data compared to us, and we get the highest average precision. But this is dubious because it's coming up with a lot of tumors, but the precision is so low. So that means an oncologist will look at that data, say, oh, we've got some tumors, let's go do these medical tests. They may be invasive, and there may be a lot of false positives in there. And so another way of looking at this, and I'm just showing that we get a large average precision. So this is another way you can look at this. So here I'm showing precision on the y-axis and recall on the x. You want an ideal line straight across the top. You want to recall a lot of tumors with the highest precision. And you're seeing the 2-pass method way in the bottom because it's not very precise. Then you're seeing in red the SSD. Then the lines above that are all of our adaptive increments. And whenever I show this to students, they always say the same thing, Dr. Toler-Franklin, that is so not impressive. But let me tell you why it's impressive, and why an oncologist would care about it. That little tiny, you see that bump that you see there? All of that are the little tiny tumors that were not being detected. What does that mean? You can detect it earlier on, you can treat, and you have a better chance of curing. And so it actually is a small object detector to detect these things early in the process. And so I just put here, this is a paper we put out just in December that deals with the other problem, the concealed object detection, where we had the camouflage spider in there. So what this paper does is it gives us a way of combining global interactions between features in our data set and local interactions well before self-attention. And so what you're seeing here is that bright blue that you see on the bottom, with that one, you can see it aligns with the detection area. But in the one on the top, which is a traditional network, vision transformer network, you don't see it detecting that tumor. And the reason is that these models in the lower layers, these are different layers of the network, these models are actually getting variations in the features early on, whereas our method allows you to find variations in the variety and types of tumors without over-smoothing later, where it's more important to do this detection. And so there's tons of future work that we're working on, and there's tons of problems in medical science that in the medical field that use neural networks, whether it's looking at databases, being more efficient, dealing with patient management, recording and interpreting data, looking at specializing primary care, cost management. We've been showing you some vision-based approaches. We're now actually looking at some physics-based approaches to doing these detections. So instead of actually detecting where the cancer tumor is, detecting the biomarkers that are used to actually trace the development of the growth of the tumor and its tumor path. And so that's dealing with quantum chemistry. And so we're looking at problems like that now. So now I'd like to talk a little bit about, so I've kind of like told you all about deep learning, how we're using deep learning in biomedical research. But now let's look at some of the motivating spectral analysis work that we've done. And I'm going to take that, show you some of the things we experimented with in our prior work, and give you a sneak peek into how we're using it on site at Tulsa. So we're focused on using spectral analysis to measure material properties. And it's motivated by this whole idea of the fact that you can image something and find out something about the actual material structure. And so this is looking at UV reflective imaging. And what you're seeing here is you're seeing two butterflies. These are cabbage-ripe butterflies that look equally bright on the left. And in the middle, even though you have the same visible light, one appears darker than the other. And this is because we put a UV filter between the sensor and the object. And so teratins, these little tiny nanostructures on that male butterfly, absorb the UV light, whereas the female on the left is reflecting it through the filter. And you get this. And here you can see they're the exact, very much the very similar color. Another effect is this iridescence, where you can look at a butterfly from different viewpoints and get different color appearance. And this is due to the fact that there are these microscopic scales on the surface and these translucent membranes that cover the scales. And what happens is that when light hits the surface, it actually, when it's reflected, is interfering with new light hitting that surface. And the slits and the size of the slits are in relationship to the size of that light, and you get this blue kind of color coming out. And we did a pilot study where we actually shadowed researchers in a number of life science fields like paleontology, forensic science, biologists, anthropologists, and we wanted to see how they were using multispectral imaging and analysis in their work to see if we could develop new ways to be able to help them in their research. And one of the things, I'm just going to pull out one of the things, is we were looking at how we can actually measure, use UV fluorescence to measure material properties. So when you're looking at these cone shells on the right in paleontology, these are completely white shells. They have no color, no information. But when you do the UV fluorescence, you see the original color patterns, and the variations in those patterns are how they do species identification. And so we were looking at cool things like this. I mean, some of the other things we looked at was the fact that in forensic science, when you're on site, you can use infrared imaging to look at heat patterns and other details that you don't see with the visible light. And so we actually put out a number of different things. We did something with microscopy. And so I'm going to focus on the UV imaging and material measurement. And so with UV imaging, you have a short wave excitation. Here you see 365 nanometers, and almost instantaneous, this long wave emission. And so that's what you see in what we call a bispectral map. You see that glowing. And from emission spectroscopy, we know that the intensity in that map is directly related to the concentration, the molecular concentration of that material. And we know that that relationship is shown in that equation where it's related to the quantum yield, it's related to the number of photons hitting, the ratio of the photons hitting the surface and being absorbed and reflected. It's related to the molar absorptivity based on which wavelength you're hitting, what that material absorbs. And so we know that there's this relationship. Another relationship is the composition. The variations in the actual hue that you get is related to the composition of the materials. And so you get this glow because these electrons are moving to this higher level. And then when they go back to rest at a lower level, you get this emission off. Another relationship that we looked at was the relationship between the composition of the materials on a surface and what you see in the hue, and the concentration and what we see in the saturation of that color. And so we're using this now. What are we trying to do with this? Well, we want to see if we can reconstruct color appearance in faded materials. And this is what we're translating onto the site at Tulsa. So here you see a material measurement system that we developed where we have ultraviolet illumination at these LED lights above an object, which is on a rotating platform. And we know that the material absorption and the emission will determine what wavelength of light we're going to use to excite that surface. And we use a number of laws related to light and the interaction with material surfaces to figure out how to actually calibrate the system to capture these patterns. So here we say UV ultraviolet irradiation at a point on a surface depends on the angle of incidence of the source and the distance from that point. And that is determining where we place these lights in our design. So what are we trying to do? So on the left, that's our shell. That's what it looks like today. It's about 3 million years old. There's no color. We want to figure out what it looked like before, and we want to use the UV to measure or figure out what the material concentration and composition is. And then we want to use our algorithms that we developed to figure out transformations that will allow us to uncover the original color. And so you're seeing that we use this ultraviolet illumination system that we developed to get B, which is a set of patterns on the surface. And then we do this really high resolution measurement with a really high powered UV radiation. And we can map it because now we can map our measurements to what we see on the surface and get a 3D texture. And so we have to deal with the contrast. So I'm showing that the emitted intensity of that spectrum will depend on the incident wavelength as well as the angle to get that contrast. So how do we use this? So here's a fun example. I thought this was a very clever trick where we measure this UV, and we have our material measurement on the surface, our colorless shell. We then look at a descendant of that shell, a modern descendant, and we, through a lot of math, figure out the relationship or the transformation between the hue and the saturation on the surface and the concentration and composition that from our measured results. And then we were able to uncover what that shell might have looked like and what the actual color patterns might have looked like. Now, the interesting thing about this is, well, you may say the modern descendant has a similar color. So the reviewers, well, they want it more convincing because we don't have a ground truth of that shell. So we did this example where we took one source, which is that eggshell there, and we're using because it has these natural materials, that one source, and then we were able to show that we were able to can reconstruct color in a broad range of different objects and materials. So here you're seeing the ground truth is on the right, our reconstruction in the middle. I love this one, the one that's in the middle there. I just love that because when my student first generated it, it was green and orange. And we got a pretty good result. That was our first good result. And so we're showing that, well, if we could do this, we know the colors of the eggs. If we can uncover this, more likely we're getting the right color here. So now I'm going to just show you, I'm going to end my talk by showing you how we want to extend this for complementing established methods in forensic science for DNA testing, mass spectroscopy, light microscopy, and electron microscopy. And so here, and also we're using the deep learning for reconstructing social networks. Okay, so let's look at the forensics. So this is the site of Greenwood at the time just before this massacre happened. It was so Greenwood is an area just outside the incorporated area of Tulsa. And it was the site of maybe, I guess it's like about 35 blocks of these really amazing homes, beautiful businesses, all African American owned businesses. And it was considered Black Wall Street. It was a very thriving community where, at the time, where there was a lot of oppression, you had a community that was feeding its own wealth back into itself and being very successful at it. But due to racial tensions in our nation, combined with triggering events on the site, in 1921, there was this massacre where the entire, there was a racial mob, and there was like this entire destruction where 1,200 homes were burnt to the ground, hundreds were looted, and 300 people are known to have died. And this is what was left. And I'm just gonna be quiet right there because it's really, really one of the biggest atrocities in our country. It was completely flattened. And so my job on this site is to put the names back to the people and to work with the forensic scientists there to bring closure to the victims who are in unmarked graves. And also I didn't go into detail about what happened, but there was fire, you have to visit Greenwood Rising. They have a really good reconstruction of what happened. There were, planes throwing fire bombs down. It was just this, it was all in like 16 hours, this entire destruction. And John and Lula Williams are a good example of a representation of the people of Tulsa at the time. So they were a very successful African American couple, very wealthy. They owned many businesses. They had a one of the two Black-owned theaters. Their economic and business portfolio included garages, a confectionery, all these businesses, and they were very successful. So today at Tulsa, the forensic scientists are working at the site. Here we're at the Woodlawn Cemetery, where they're actually uncovering and trying to find the mass graves. They are using DNA testing to identify the bodies, and then they're giving them proper burials and returning them to their families. And it is a very important community event, and it's meant to be an activity to bring closure because the local community is involved in actually escorting the remains and being involved in volunteering on the site and helping with the excavation, because typically these types of excavations are exclusionary. And so now they have an ability to take a part in it. And so this is the team from the first site, and I work with the lead forensic scientist there, Phoebe Stubblefield. We have worked for many years. This is after a long day on the site, but we work together for many years. I've been working at her. She runs the C.A. Pound Human Identification Lab at the University of Florida, and I've done a lot of things in laboratory settings with her, like imaging gunshot wounds with spectral imaging and other things. And so this was great to be able to work with her on site. And so here you see what the excavation site looks like at a potter's field here. One of the things Phoebe was really surprised at was the order in which these sites were laid out. Typically, if you think of a potter's field, you may just think of bones being buried without any formal burial. And one of the interesting things on the site is they're actually reconstructing and mapping out what the burial ground looks like as they uncover. So one of the things they're using on the site for human identification is DNA testing. So what the first victim was identified through these means in 2024. C.L. Daniel was a veteran who served our country in World War I, who was killed in the massacre, and whose family did not know where he'd been buried for the last 103 years until that week. So what are some of the challenges? So one of the challenges with the DNA testing is that it's destructive and sometimes equivocal. And if there's sparse data, like these remains that have been around for so long, you may not get any results. And also privacy is an issue. You need samples from descendants in order to be able to do this. And so this is on site. This summer, I spent time there. This is Brenda Nails Alford is on the left. She is a descendant of one of the survivors of the massacre, and she works on the site with Phoebe. Phoebe Stubblefield is the center, and I'm on the right. And so what we're doing is we're looking at ways that we can use the spectral imaging and deep learning technology to enhance what has been done with the DNA testing. And so here we're looking at looking at the grave markers and the headstones. And so these are the challenges. So you have unmarked graves, you have headstones that don't have any legible text on them. You have situations where you may have weathering or other types of destruction from the fire that happened that destroyed this information. And so we're trying to figure out ways to combine and use complementary methods. Another example here, this is actually a sample of cloth. And being able to identify the cloth, being able to figure out why the cloth was there and what role the cloth played, or who, it also helps identify who is in the particular burial. And so here's an example of another piece of blue cloth that they're looking at. So on site, they use some traditional methods like mass spectroscopy, looking at using these techniques to look at the ionized gas and figure out what is the mass. I just highlighted methods that they use on site. What is the weight, the molecular weight, the structural characterization, and to do qualitative and quantitative analysis of what's there. And so some of the challenges with this is destructive. you need certain expertise, and it requires certain resources and collaboration. They also do light microscopy. So just looking at visible light through a lens and being able to magnify and look at the details on the site, the details of the particular specimen. And so this can be destructive, and it's limited to this visual inspection. We want to do some analysis. And so another method is scanning electron microscopy, where you shoot electrons at a sample, and because you're shooting at this quantum level, you're getting this really high resolution detail, and you can look at the molecular structure, the anatomic makeup of the specimen. And so this is another method that they use. So the projects that I showed before were the first inspiration behind methods we're developing now to complement or even replace some of these methods. Another challenge that we look at is the fact that the death certificates on site, because we want to link what's on the site to public records. The death certificates have cities and places that no longer exist, diseases that no longer exist, conditions that no longer exist, not to mention the fact that the females, a lot of the women on the site may not have been recorded correctly, or may have been recorded with the names of their husbands. I mean, it's a really interesting thing. So we're looking at neural networks that actually do this multimodal reconstruction and be able to match with these records. When you think about today, we all have social networks. You use your phone, you're on social media, you're sitting here with me today. How do you reconstruct a social network that once existed that is sparse, where the people have migrated, where the whole community has been destroyed, where information is missing? These are the challenges that we want to apply AI to. And this just shows the death certificates from Tulsa, from about the time, you can see the spike at the time of the massacre, and you're seeing the numbers of death certificates and the male and female examples from time. So we're on site, we're looking at developing the system that combines spectral imaging, material analysis, and AI for reconstructing this social network. And so this system will address rights issues for individuals marginalized in death by providing mechanisms to reconstruct history and identity. The results will bring equity to the importance of death records in African American communities where deaths are underrepresented. And that's it for me. And I want to thank all the people that I work with.
Microphones are on the way. So if you have a question, please raise your hand. We'll get your microphone right away.
Oh, thank you.
Right. Give it a second.
Is it working? So actually, I work in mass spectrometry, and we're trying to analyze some gene therapy products. And one of the things that we have seen in our software, it's commercial software, right? So I never sure whether to believe it or not. Okay, and yeah, what we have is something called a false discovery rate, which means we put in a decoy to tell us if it's correct or incorrect, okay? And those decoys for peptides, for their molecular weight, can be arranged in either a random sequence or a inverted sequence or simply a scrambled sequence. And so it comes up because we're trying to insert a gene for human structure into a monkey, and we're trying to differentiate the monkey and the human. How do your systems have like decoys in them to tell you that you're looking at something, you believe it, but it's wrong?
Right.
That's
So, okay, so first, I'm not an expert in mass spectrometry, right? But we know the mechanisms by which they work, and we try to make analogs to them. So far in our work, and this isn't out yet, but so far what we do is we don't actually have a decoy. Rather, we have computational methods that give us a prediction of what the actual molecular structure is. And so far, we've just been, so far we know what the answer is, and we've just been comparing our results to what the answers are to see if it's correct. But we don't have like a decoy or a complex system in that way. So right now we're just testing if we can, we're just seeing if we can do the right identification right now.
It was a great talk. Thank you very much. I have a twofold question. So there was a shell example that you showed, which had absolutely no color, thousands of years ago, or, whatever the time frame was. You started how it looks like today, and then with the methods, you reconstructed how it would look today. The question is, so you must have gone through many iterations of maths or whatever the algorithms are. How do you know that that's how it must have looked? You could have, you could have been completely off and completely right. So, number one, how was that unsupervised learning of the algorithms, or you actually supervised, hey, this direction is not right, need to go in a different direction? And then finally, how did you decide the authenticity of the look finally that you arrived at?
Right. So our methods did not, they were not supervised. And well, let me say another. So first of all, what we were looking at is, so when we got the modern descendant of that particular specimen, we were looking at it from the standpoint of what is the mathematical relationship that we see in spectroscopy between the color and the way the color is distributed on the shape and the molecular density and material composition. And so we were looking at just modeling that relationship. And the idea we had is that if we could model that within anything of the same material, we got something else of the same material, we should be able to model it. Now we know what those shells are made of, so we have to just make sure that we have the same material. Now, in terms of the authenticity of what we reconstructed, nobody knows, because some of the shells are 65 million years old. Nobody knows. And that was the biggest criticism that reviewers of the paper had. And so when I resubmitted, the best we could do is take samples of different materials like the eggshells that I showed and demonstrate that for cases where we do know the answer, we got the right answer. And that was what I had to do to convince them, because nobody knows. And so, but by showing on a broad range that just with one sample, whether it's purple, green, red, or whatever, that one sample, we could get a broad range of density, complexity, and color. That's what we had to do to convince. But yeah, I don't have an answer of what it really looked like. We're just telling people what we see. But the concentration, stuff like that, I mean, you could measure that. I mean, that's pretty, yeah, you can get the accuracy of that. It's the color the color parents that you're not sure of.
Hello. Dr. Toler-Franklin, great presentation. My question is on the tumor research. Now, my knowledge of AI, it comes from the language models of OpenAI and DeepSeek articles in the news.
Yes.
And they always talk about the size of the model in terms of parameters, 300 billion parameters versus a smaller model that has 10 billion parameters. I was wondering whether that is a fair question in your work,
it is.
and how many parameters are there?
Yes. So this model is actually not that big of a model. Yeah, so the model size is completely important. Obviously, the larger the model we have, the more accuracy we get. But then so we had a trade-off between model size and being able to do this efficiently and quickly. So it's the same thing. I mean, I would think it's not as large as a natural, nowhere near. So you could think of this as a tiny model compared to your world. But yes, it's a fair question, and it's something that is really important because even the second cancer tumor paper that you saw came out 2024, we had to when we to the archive that's out is different from what's being published because we then needed to do some pre-training and see how the pre-training numbers added up. And then we had to be very careful that we had the same number of parameters, because when we do the ablation test, we have a base vision transformer. We have our transformer, which has this aggressive convolutional pooling. It also has this conceptual transformer attention, right, in hyperspace. So we had to make sure that all the same parameter sizes of the base were the same as this when we did our comparisons. So these are things, yes, we have to be very careful of and to make sure it's
Thank you.
consistent.
Here we go.
So forgive me if you discussed this already, but when you were talking about the deep learning between the supervised and the unsupervised, and you were essentially discussing the biomarkers for the cancer, do you start with known biomarkers in the supervised realm, and then you use that to then translate to the unsupervised? Is that how that works?
So all of the results I showed today was from a vision perspective, where we were just using the models like a vision transformer to detect the actual tumor. I did not talk about the actual work that we're doing with the biomarkers. That's more of a chemistry problem. And yeah, so that one's a little different. Um, and um, and so the first tests with that are supervised to see if you can find the expression, what protein is being expressed. But yeah, I actually didn't talk about that work. That work's not published yet.
So when you were even showing that analysis where you had all your plot points and all that kind of stuff, how do you determine what's how does the detection system then determine what's
okay,
noise versus not
Okay,
noise?
so for the vision one, the one that is labeled. So the oncologist has given us a set of they get the imperial slide, they have like annotation software, researchers in the lab actually annotate where the tumors are, he confirms it, then he gave us a whole big data set of that, and we trained our model on that data set, then given a new data set we could detect. So that was a label problem.
Thank you.
I have a question before we put up the mic. I have a quick question for you. So you put up a slide that on this one where you showed a very large tumor and then some very small tumors, and to the point where, at least to our untrained eye, it was getting harder and harder to tell. I have a friend who's a radiologist, and we have this debate all the time. But given your work in AI and visual recognition, how quickly do you expect to see AI deep learning really start to move into radiology?
It already has, and every time, so I teach a course, where I taught a course for several years with the medical honors students at the University of Florida. And the first thing they come in with their sad faces is like, especially the radiologist, is my job going away? And I tell them, no, you're going to be a new informed type of user of these things. So it is there. So basically, there are a number of standards now set out. There are a number of like federal standards even for like parameterization for these models. There are some hospitals that were involved in some large scale projects where they're actually using radiology to detect things like, oh, they're detecting break a bone breakage and things like that. So there are research labs and at hospitals that do this today. And yeah, and I think the thing that shocked me, I think like especially the last time I taught, between every time I finish the class, I tell them here's what we need to look forward to in the future, several years down. The last time I taught the class, everything that I projected and all my wisdom about what would happen later had happened in that year with a number of startup companies. So it's very fast and it's in all areas. It's everywhere. Yeah.
Thank you. Thank you. I saw microphones. Oh, there we go.
Oh, thank you. Yeah, I have a question about the use of this technique in cancer detection. So the oncologist takes a biopsy, sends it to a pathologist who meticulously and looks at the slide, looking for the certain markers, cells, et cetera. Is the AI, would it be used as a pre-processor? Because the AI is not 100% accurate. So what, it would be an assist for the pathologist, or would it be a
Okay, so there's two pathways. For the vision transformer pathway, the idea is that it's an assist for the oncologist. When we think about the biomarker work, that's where it gets interesting because the pathologist is now nervous because the argument we make, and the pathologist always corrects me, I say that it's subjective. No, he says it's somewhat subjective because they look at the margin they look at the percentage of tumor expressed, and they look at the margins and they use a morphological software to figure it out. And so in that case, we want to do what the pathologists do more accurately, and then basically we want to do it, we want to test it against standards that exist, and then we want to be able to have the pathologist be a part of the process to further verify, but we kind of want to take away a little bit of the subjective nature of the visual inspection and so that's where we want to get to. We're not anywhere near there yet, but that's where we're going for.
To your right, there's a student right here.
Um, have you done any spectral analysis with like astrophotography and on like pictures of nebulae and seeing the different gases?
I haven't, but I've been on the committee of a cool physics student who was looking at problems like that. But no, I know exactly what you're talking about because I've been on this committee where we're looking at dust bursts and the changes and the fact that the infrared lights and how they express. But no, I personally have not done with physics, with astrophysics or looking at nebula.
And with that, I think no more questions. Could we please thank Professor Toler-Franklin once again for a wonderful
Thank you
talk?
for coming.